Hello, my name is Vasundhara Agrawal, and I'm the Applications Manager for Spatial Bioimage Analysis at Leica Microsystems. Studying a complex interplay among tumor, stromal, and immune cells within the heterogeneous tumor microenvironment necessitates a multiplexed analytical approach to investigate cancerous tissues from diverse origins to ultimately predict clinical outcomes and design novel therapies. Therefore, in this study, we analyzed a large panel of cell signaling technology antibodies that are targeting various cell types using immune, epithelial, and vascular markers in cancer tissues derived from various origins, including lung, colon, and pancreas. The TME for such cancer types may show differential immune reprogramming, revealing unique characteristics and molecular clusters, their interactions based on the tissue type. CST antibodies undergo a vigorous validation process to ensure antibody performance on FFPE tissues. All antibodies in this study were direct conjugates and are shown in Table 1 here. These antibodies were then used for subsequent staining of various cancer tissue types, shown here. Slides were imaged on the cell dive imager using 4 channels plus DAPI with automatic autofluorescence removal, corrections, and stitching. Fully stitched images were then analyzed using Aivia 13.1. If we look at Figure 1, we can see the final multiplex cell dive imaging in 4 cancer tissue types. A shows mucinous cyst adenocarcinoma of the ovary. B shows pancreatic ductal adenocarcinoma. C shows large cell lung carcinoma. And D shows colon adenocarcinoma. Now, moving towards Figure 2, using AI-driven analysis, the expression of markers shown in Table 1 was characterized through autoclustering. Simply based on biomarker expression patterns, clustering of different cell populations is determined in each cancer tissue type in a non-biased method to determine interactions that are tissue-specific and those that are common to all cancer tissue types within the study. For example, in ovarian mucinous cyst adenocarcinoma, clustering reveals unique lymphoid cell populations, such as cluster 19, shown here, expressing CD56, CD45, along with expression of exhaustion marker TIM3. Furthermore, cluster 10, which is right next to cluster 19, indicates a diverse myeloid cell population with potentially immunosuppressive and tumor-promoting effects. Similarly, other unique clusters were identified in other cancer tissues. For example, cluster 10 in pancreatic ductal adenocarcinoma is indicative of regulatory T cells and also co-expresses immune checkpoint molecules. Cluster 19 in large lung cell carcinoma expressing CD45, PD1, CD3, CTLA4 indicate T cell expressing immune checkpoint markers, again suggestive of immune evasion. And cluster 1 in colon adenocarcinoma expressing vimentin, CD11C, CD68, CD163, and TIM3 potentially indicates a subset of TAMs, suggesting a regulatory phenotype contributing to tumor growth and immunosuppression. Now, in Figure 3, we can see the multiplex cell dive images of select clusters and unique cell populations identified in the 4 tissue types studied here. So to summarize, clustering analysis of multiplex cell dive images gives us insights into the unique cellular composition and immune microenvironment of each cancer site, highlighting potential prognostic implication and therapeutic targets. If you'd like to take a closer look at this poster, it is available for download. Thank you for listening.